Good morning. Today we will study the lectures of the tense Golgi body or Golgi apparatus. The Golgi body is made up of stack of membrane-bound bodies known as the cisterna. About discovery of the Golgi apparatus. The Golgi apparatus is noticeable with both light and electron microscope. It's also called Golgi complex, Golgi body, or simple Golgi. The Golgi complex was discovered by Italian physician and Nobel laureate Camille Golgi in 1898 during an investigation of the nervous systems. Its electron microscope structures was discovered by Dalton and Felix in 1954 years. Location of the Golgi apparatus to the cell. The Golgi apparatus is present in all eukaryotic cells and absent in prokaryotes. The Golgi apparatus is located in between the endoplasmic reticulum in the cell membrane. Most often, the Golgi appears to be extension of the endoplasmic reticulum, which is slightly smaller and smooth in appearance. However, the Golgi apparatus can be easily mistaken for smooth endoplasmic reticulum. Also, they look similar. The Golgi is an independent organelle which has a different function. Main function of the Golgi body. First function, a cell may have an own large complex or several very small ons. It occupies different positions in different kinds of cells. Second function is secretory and absorptive cells is usually least between nucleus. Third question, the invertebrate and plant cells usually have several small Golgi complex called dictyosomes scattered over the cytoplasm. Structure of Golgi complex. The main first structure of Golgi complex varies in size and form in different cell types, but usually has similar organization for any type of cells. Second structures consist of 4 functionally distinct regions: entry phase, middle cisterna, trans cisterna, and exit phase. And the structure, each region contains different enzymes which selective modifies the contents depending on where they reside. What does it mean of the cisterna? It's the simplest unit of the Golgi apparatus in the cisterna. Cisterna, about 1 nanometer in diameter, are central, flattened, plate-like or saucer-like closed compartments that are held in parallel bundles or stakes on above the other. In each stake, cisterna are separated by a space of 20 to 30 nanometers, which may contain rod-like elements of fibers. Each stake of cisterna forms a dictosome, which may contain 5 to 6 Golgi cisterna in animal cells or 20 or cisterna in plant cells. The next slide, you can see the structure Golgi apparatus, what are including transport vesicle, secretory vesicle, and surrounded by cell membrane. Golgi apparatus included tubules. What does it mean, tubules? Tubules is small round tubules arise from periphery of the cisterna. Some of the enlarge at the ends to form vesicles. Vesicles. What does it mean, vesicles? Vesicles lie near the ends and concave surface of the Golgi complex. There are 2 types: smooth or secreted vesicles, and coated vesicles. All the Golgi elements are filled with a fluid, the Golgi matrix. Function of Golgi complex. The Golgi complex, including the next main function. First function, modifies proteins and lipids. Second question, functions, make and secretory mucus. Third function, package products into vesicles for transport. On the slides, you can see the process transport of the packaged products into vesicles for transfer to the ER or endoplasmic reticulum. Golgi apparatus consists of cisterna, lumen, cis phase, transport vesicles from ER. The Golgi apparatus and the endoplasmic reticulum or ER work together to produce new cell membrane. The Golgi also creates lysosomes. These sacs contain digestivate materials. The lysosomes are pinched off from the Golgi apparatus and they are used to process materials which have been photosynthesis or digest organelles which no longer functions. The lysosome delivers raw ingredients to the endoplasmic. On the slides, you can see the process forming of the primary lysosomes. It's a fusion form secondary of the lysosomes.
Often found near the endoplasmic reticulum inside the cell, the Golgi body is a cell organelle that's in charge of packaging and shipping lipids and proteins. As a part of the endomembrane system, Golgi bodies take existing protein and lipid structures that have been synthesized in the endoplasmic reticulum and modifies them. Let's take a look at the basic workflow of the Golgi body. Proteins and lipids arrive at the Golgi body in packages called vesicles. Vesicles are a basic membrane that is used to enclose materials in order to sort and protect cargo during transportation. Once proteins and lipids arrive at the Golgi body, the Golgi apparatus modifies them by changing their molecular structure. This effectively prepares the proteins and lipids to be able to perform whatever function they are designed to carry out. After the molecules have been modified, the Golgi body packages them into vesicles again and ships them to either a place inside the cell that has a need or to the boundary of the cell where they are expelled to be used outside the cell.